Alright everyone, we are down to the last topic of our midterms, and welcome to Trematodes. Other group of flatworms is composed of the FLUKES or Trematodes. Adult Trematodes are equipped with an oral sucker and the ventral sucker called an acetabulum. A third sucker called a genital sucker or gonotile is observed only among the heterophytes. They are all hermaphroditic. All trematodes require two intermediate hosts in their life cycle. All trematodes have operculated eggs, and the infective stage for all these trematodes is the encysted larva. The metacercaria that develops in the second intermediate hosts. These characteristics are observed in all medically important trematodes, with the exception of the cystosomes, in which the infective stage is the cercaria. While the first intermediate host is always a snail, the second intermediate host may be a fish, crustacean, another snail, or freshwater plants. For our objectives, so at the end of the course, the second year Bachelor of Science in Medical Laboratory Science students are able to first, enumerate different types of flukes according to their location in the human body. Second is to describe the morphology and the diagnostic stage features of each fluke. And lastly, identify the infective stage, the mode of transmission, diagnostic features of each parasite. Okay, before we formally start with our discussion, I want you to remember some of these. The eggs of our trematodes are operculated except for Schistosoma species. All trematodes require secondary intermediate host except for Schistosoma species. The infective stage is metacercaria except in Schistosoma species, which is the fork-tailed cercariae. The mode of transmission is via ingestion except in Schistosoma species, and then the first intermediate host for our trematodes is the snail, and the secondary intermediate hosts are the snail, plant, fish, crab, and insects. Now everyone, I want you to remember that the trematodes pass through three morphologic forms during their life cycle. So the first thing is the egg, the second one is the multiple larval stages, and lastly, into an adult worm. The eggs, which are the primary morphologic form recovered in human specimens, vary in appearance. Some contain a lid-like structure that, under the appropriate conditions, flips open to release its contents for further development called an operculum, such as in Fasciolopsis and Fasciola. Other members of the trematodes may be distinguished by the presence and location of spines, as seen in the Schistosoma species. The various larva stages typically occur outside the human host and are rarely, if ever, encountered. The rarely seen adult flukes are thin and non-segmented, which resembles leaves in shape and thickness. They typically range in length from 1 to 5 centimeters. Each adult fluke is equipped with two muscular cup-shaped suckers, one oral and the other located ventrally, a simple digestive system, and a genital tract. Like the typical cestode, the average trematode uses its body surface as a means for absorbing and releasing essential nutrients and waste products. For classification purposes, the flukes or trematodes may be divided into four categories based on the areas of the body that primarily harbor the parasites, which includes the intestine, the liver, the lung, and the blood. Thus, they are called as blood flukes, liver flukes, intestinal flukes, and lung fluke. Upon knowing the four categories of our trematodes, Now we are going to discuss them one by one. Let us start with the blood flukes, or also known as your cystosomes. Starting with our cystosomes or cystosoma. Cystosoma is a genus of parasitic blood flukes that infect birds and mammals, including humans. There are 5 species of medically important cystosoma have been identified, such as Cystosoma japonicum, Cystosoma mansoni, Cystosoma hematobium, Cystosoma mekongii, and Cystosoma intercalatum. Cystosoma japonicum is the predominant species in the Philippines. Cystosoma japonicum, or the oriental blood fluke, causes Schistosomiasis japonica. It is endemic in China, the Philippines, and Indonesia. It was first described in Japan, but has been eliminated, with the last human case reported in the year 1977. The strains of Cystosoma japonicum from the different geographic regions are genetically distinct, but all require snails of the species Oncomillania as their intermediate hosts. So as you can see in the picture, this is a Cystosoma japonicum parasite in their adult forms, both the male and female, which are in copula. Now let us proceed to the adult worms of our Cystosoma japonicum, starting with the male adult worm. It is described as elongated, cylindrical in shape, measuring about 12 to 20 millimeters by 0.5 to 0.55 millimeters in diameter. The integument is non-tuberculate, gynecophoric canal is present. The oral and ventral suckers, as you can see in the picture, lie close together at the anterior end, with the ventral sucker as a shallow cup suspended from a short base. The alimentary system consists of the mouth, surrounded by the oral sucker, the esophagus, then the intestine, which bifurcates in front of the ventral sucker and joins again in the last fourth of the body. The male adult worm has 7 medium ovoid slightly lobate testes in single column near the anterior end of the worm, each provided with a short vas deferens emerging into the seminal vesicle before opening into the exterior through the genital pore. Next is the female adult worm of our Cystosoma japonicum. It is described as more slender and delicate than the male adult worm, and it measures about 26 millimeters by 0.3 millimeters in diameter. The ovary lies somewhat behind the middle of the body in front of the union of the intestinal ceca, and then emerging from the ovary is the oviduct, which bends forward and run parallel to the vitelline duct. The female worm is lodged in the gynecophoral canal of the male worm upon copulation. So, if you are having difficulty of identifying and locating the various parts of our Cystosoma japonicum male and female adult worms, you can refer to this picture as your reference. Now let us proceed to the ovum or the egg of our Schistosoma japonicum, which is described as ovoid, round, or pear-shaped if you are gonna refer to our picture on the right side, and it is somehow pale yellow in color. The longer diameter ranges from 46 to 110 micrometers, while the shorter diameter of the egg ranges from 37 to 90 micrometers. It has a thin shell onto which residual tissue or red cells may be adherent. A curved hook or spine, as you can see in the red arrow at our picture, may be observed near one of the polar ends, but only if the egg is properly oriented. So on the side near one end is a depression from which extends a small spinos. And then the Schistosoma japonicum egg is embryonated when laid, and it has a ciliated miracidium with attenuated anterior end creating the sloping shoulder within the shell. This is an actual image taken by me of Schistosoma japonicum egg under the scanner objective. This is how it looks like under the low power objective. And this is what it looks like under the high power objective. Showing you a picture of Schistosoma japonicum egg with some of its labeled parts. So the next one is the Schistosoma japonicum sporocyst, which is a thin-walled saccular structure filled with germ cells. The sporocysts develop from a miracidia, which is seeking out and infecting the snail intermediate host, which is Oncomillania hupensis quadrasi, and sporocysts are able to reproduce asexually and can later give rise to free-swimming cercariae after 60 to 70 days. The next one is our cercaria or cercariae, which is described as elongated, pear-shaped body with rounded end and a long forked tail, as you can see in the picture. The main body measures from 100 to 500 micrometers in length and 40 to 60 micrometers transversely. The tail trunk is 140 to 150 micrometers by 20 to 35 micrometers, and the forked tail is measuring about 50 to 70 micrometers long. So the tail is characteristically bifid, and then the cuticle of the body and tail is covered with minute spines except at the region of the anterior sucker and tips of the tail. So the cercariae penetrates the skin of the definitive host when the host comes into contact with infested fresh water. The cercariae then lose their forked tails and transforms into a schistosomula and enter the superficial lymphatic vessels or the subcutaneous veins and reach the lungs of the host. So the treatment or the drug of choice for our Schistosoma japonicum is praziquantel. This is an actual image taken by me of Schistosoma japonicum cercariae under the scanner objective. This is how it looks like under the low power objective. Now let us look closely under the high power objective. This is also an actual image taken by me of Schistosoma japonicum miracidium under the scanner objective. This is what it looks like under the low power objective. And this is how it looks like under the high power objective. And we are done with our Schistosoma japonicum. Let us proceed with the next schistosome, which is the Schistosoma mansoni. Common name is the Manson's blood fluke. The habitat is our inferior mesenteric vein of the colon. Intermediate host is Biomphallaria. Location of the ovary is anterior part of the adult worm, which is one half of its body. And then egg production is 190 to 300 eggs per day. And the number of testes is 8 to 9. Let us discuss the male adult worm of our Schistosoma mansoni. It is elongated, cylindrical in shape, measuring about 6 to 14 millimeters by 1.1 millimeters in diameter. Integument is tuberculate, and then the intestinal ceca unite early in the anterior half of the body. United intestine is long. The testes are small, so 8 to 9, and are zigzag row. Other structures is somewhat similar to our Schistosoma japonicum. Let us proceed to the female adult worm of our Schistosoma mansoni. It measures 14 millimeters by 0.25 millimeters in diameter. The uterus is short with usually 1 to 3 eggs, and then the ovary is located in the anterior half of the body, and some of its structures is similar to our Schistosoma japonicum. The ovum or egg of our Schistosoma mansoni is ovoidal in shape, measuring 155 by 60 micra, sometimes with S-like curvature. It is yellowish-brown in color, transparent shell with a characteristic lateral spine as you can see in the picture, and the egg or the ovum is embryonated when laid with isolated miracidium. This picture shows the egg of Schistosoma mansoni with some of its labeled parts. Now we are down to our last schistosome, which is the Schistosoma hematobium. The common name is the bladder fluke, and the habitats are the following: vesical area, prostatic area, uterine plexuses of the venous circulation. The intermediate host is the genus Bolinus. Location of the ovary is in the posterior area, which occupies one half of its body. And the egg production is 30 eggs per day, and the number of testes is 4 to 5. Let us proceed to the male adult worm of our Schistosoma hematobium. It is described as elongated, cylindrical in shape, measuring about 15 to 19 millimeters by 0.8 to 1 millimeters in diameter. Integument is covered with minute integumentary tuberculations or fine tuberculate, and then the intestinal ceca is long. reunite at the middle of the body. The intestines are in groups, 4 to 5 in number, found just behind the ventral sucker, and other structures are similar to Schistosoma japonicum. Next one is the female adult worm of our Schistosoma hematobium. It is described as long and slender as compared to the male adult worm of our Schistosoma hematobium. The female adult worm measures 20 millimeters by 0.25 millimeters in diameter. The uterus has 20 to 30 eggs at a time, so the suckers of the female adult worm are somehow small, subequal, and conspicuously muscular. The ovary is in the posterior half of the body, and other structures are similar to our Schistosoma japonicum. As for the ovum of our Schistosoma hematobium, it is described as elongated, ovoidal in shape, and it measures 112 to 170 by 50 micra. It is light yellowish brown in color and has a transparent shell as you can see in the picture, and it has a distinct terminal spine. So it is partly mature when laid with miracidium, and in the tissues the eggs appear to be spindle shaped with rounded anterior and conical posterior end with a terminal spine. This picture shows the egg of our Schistosoma hematobium with some of its labeled parts. This table shows the summary of the different blood flukes or schistosomes. Now class, I want you to familiarize the different characteristics of each parasite. This picture shows the various blood flukes or schistosomes with their distinguished features of the ovum. And we are done with the different blood flukes. Now let us proceed to the liver flukes. Fasciola species are large digenetic trematode species belonging to the family Fasciolidae. They are parasites found in the liver and biliary passages of humans and herbivorous mammals, especially ruminants. Fasciola hepatica is also known as your temperate liver fluke, and Fasciola gigantica is also known as your tropical liver fluke, are the causative agents of fascioliasis. Now let us discuss them one by one. Let us go first with the adult worms of our Fasciola hepatica and Fasciola gigantica. The adult worms are large, flat, brownish, leaf-shaped, and it measures 3 centimeters by 1.5 centimeters, in which Fasciola gigantica is longer than Fasciola hepatica, as you can see in the picture. It has 2 ventral suckers, in which the ventral sucker of our Fasciola gigantica is larger than Fasciola hepatica, and then the anterior end has a conical projection called the cephalic cone, giving the worm a shouldered appearance, in which Fasciola gigantica has a shorter cephalic cone than Fasciola hepatica. The integument is covered scales, but posterior surface may be smooth. Alimentary system has an inverted Y appearance. The branch intestinal ceca bear a number of lateral compound branches, and the genital system is similar to other trematodes. Now let us go specifically to our Fasciola hepatica adult worm. Fasciola hepatica has a large, broad, and flat body which measures 18 to 51 millimeters in length and 4 to 13 millimeters in width, which is near the mid-body. A distinguishing feature is the cephalic cone, which has a marked widening at the base of the cone, or also known as the shoulder, as you can see in the picture at the left part of our PowerPoint. So the suckers are comparatively small and are located close to each other in the conical projection. Next is our Fasciola gigantica. So as compared to Fasciola hepatica, Fasciola gigantica adult worm is longer, which is 25 to 75 millimeters, with about the same width of 3 to 12 millimeters, with less developed shoulders and a shorter cephalic cone. The ceca are more branched, especially towards the midline of the body, and the branches of the ovary are longer and are more numerous as compared to our Fasciola hepatica. Okay, so now let's proceed to our Fasciola species egg. Fasciola hepatica and Fasciola gigantica eggs are effectively morphologically indistinguishable and also can be difficult to distinguish from or can be confused with eggs of Fasciolopsis pusci and eggs of some Echinostomus species. The Fasciola hepatica egg is large, ovoidal, operculated, and yellowish to brownish in color. It measures 140 to 180 micrometers by 63 to 90 micrometers in size and is released from the worm still immature, containing a large unsegmented mass of vitelline cells, while the Fasciola gigantica egg is slightly larger than the Fasciola hepatica egg, which measures 160 to 190 micrometers by 70 to 90 micrometers. This is an actual image taken of a prepared slide of Fasciola hepatica under the scanner objective. And this is what it looks like under the low power objective. And let's take a closer look of our Fasciola hepatica under the high power objective. Now we also have an actual image of a prepared slide of our Fasciola gigantica under the scanner objective. This is what it looks like under the low power objective. And this is how it looks under the high power objective. The next one is the miracidium or the miracidia of our Fasciola hepatica and Fasciola gigantica. It is described as free-swimming ciliated 130 by 27 micra in size, and it has a cephalic papillae, eye spots, 2 flame cells, and a rudimentary digestive tract. Inside the snail, the miracidium develops into a sporocyst, followed by 1 or 2 generations of rhidiae, which produce cercariae. And lastly, we have the sporocyst of our Fasciola hepatica and Fasciola gigantica, which develops from our miracidium or miracidia. The sporocyst is described as a thin-walled saccular structure filled with germ cells, as you can see in our picture on the right portion of our PowerPoint. The treatment for our Fasciola hepatica and our Fasciola gigantica is triclabendazole. Now let us proceed to our next liver fluke, which is Clonorchis sinensis, or also known as your Chinese liver fluke. The liver flukes such as your Clonorchis sinensis, Opisthorchis filineus, and Opisthorchis viverini have similar life cycles. They usually usual mode of transmission is via ingestion of the metacercaria of the parasite present in infected raw or undercooked fish. Usually, the adult worm of our Clunorchis sinensis is described as narrow, oblong in shape, flat, tapering anteriorly, and rounded posteriorly. So it measures 10 to 25 millimeters by 2 to 3 millimeters wide. The oral sucker is larger than the ventral sucker. The ventral sucker is situated at the junction of the anterior and middle third of the body. The simple line intestinal ceca extends to the caudal region of our adult worm, and then the large testes, which are deeply lobed and found in the posterior third of the body, one behind another. Now let us proceed to the egg or the ovum of our Clunorchis sinensis. So the eggs of Clunorchis sinensis is somehow yellowish brown in color, ovoid, and it measures 26 to 30 micrometers by 15 to 17 micrometers. There is a distinctly convex operculum that fits into the thickened rim of the shell, as you can see in the lower part of our PowerPoint or in the picture, and the small protuberance at the abopercular end of the egg. Inside the egg is a well-developed miracidium that has asymmetrical features, and the eggs of the three species of our liver flukes are very difficult to differentiate. The treatment for our Clunorchis sinensis, or the drug of choice, is praziquantel. This is an actual image taken by me of our Clunorchis sinensis under the scanner objective. This is how it looks under the low power objective. And let us take a closer look of our Clunorchis sinensis under the high power objective. This picture shows our Clunorchis sinensis egg with some of its labeled parts. And the last liver fluke that we are going to discuss is our Opisturkis viverini, or the fish liver fluke. So Opisturkis adults are slightly shorter, being 8 to 12 millimeters long and 1.5 to 3 millimeters wide. The main similarity between Clunorchis sinensis and Opisturkis species is the location of the vitellaria, which are found in the middle third of the body at the level of the uterus, whereas the main differences are in the morphology and arrangement of their testes, in which the adult worm of Opisturkis viverini, the testes are lobate and it is situated in the posterior fourth of the body. So let us describe some more of our Opisturkis viverini adult worm. It is flat, elongated, aspinous, and has a tapering anteriorly and rounded posterior shape. Now let us proceed to the ovum or egg of our Opisturkis viverini, which is described as oval-shaped with an operculum resting on shoulders. It measures 26 by 30 micra, and it may or may not possess terminal tubercle-like knob. And always remember that our Opisturkis viverini are fully embryonated when laid. And the treatment is also praziquantel. This table shows the summary of our liver flukes, and I have also included some nice to knows about Opisturkis felineus and your Fasciola dendriticum. Now let us proceed to our intestinal flukes. The fasciolate digenetic trematode is the largest intestinal fluke of humans and pigs. So the mode of transmission of our Fasciolopsis busci is by ingestion of incested metacercariae from aquatic plants. This can occur when the plant itself is eaten or when the hull or skin of the fruits of these plants is peeled off between the teeth. Now let us describe to the adult worm of our Fasciolopsis busci, or also known as the giant or the large intestinal fluke. It is described as large, broadly ovate or oval. Anterior end is narrower than the posterior end. It measures 2 to 7.5 centimeters by 0.8 to 2 centimeters. The ventral sucker is larger than the oral sucker, and it somehow resembles our fasciola hepatica, but it does not possess any cephalic cone. The alimentary tract is simple, but the ceca do not bear any lateral branches. The next one is the ovum or the egg of our Fasciolopsis busci. So the egg is large, operculated, and embryonated when first passed in the feces, and it is indistinguishable from the eggs of Fasciola hepatica and Fasciola gigantica. It measures 130 to 140 micrometers by 80 to 85 micrometers. And the egg is somehow yellowish brown in color. It is also ellipsoidal, and it has a clear, thick shell with a small operculum at one end. And the treatment used for this parasite is praziquantel. This picture shows the egg or the ovum of our Fasciolopsis busci with some of its labeled parts. Now let us proceed to our next parasite, which is the Echinostoma elucanum, or also known as your Garrison's fluke. The echinostomids are digenetic trematodes characterized by a collar of spines around their oral suckers. There are several species which infected humans. 2 species have been documented in the Philippines, and that would be your Echinostoma elucanum and your Artifechinostomum malayanum. Let us describe our Echinostoma elucanum adult worm. The Echinostoma elucanum adult worm is somehow reddish gray in color, and it measures 2.5 to 6.6 millimeters in length and 1 to 1.35 millimeters in width. The oral sucker is smaller than the ventral sucker. Simple intestine of an equal length which extend to the subcaudal region, and then it has 2 deeply lobed testes which lie one behind the other in the middle of the posterior body, behind the globular ovary, and then the vitellaria are medium-sized follicles in the lateral fields through the posterior 3 ports of the body. Next is the egg of our Echinostoma elucanum, which is straw-colored, operculated, and ovoid, which measures 83 to 116 micrometers by 58 to 69 micrometers. Next is the iridia of our Echinostoma elucanum. So it is described as elongated structure which has anterior collar and a rhabdi cell, gut, and a locomotor, and it contains 3 to 20 daughter iria inside of it. Next is the cercaria or the cercariae of our Echinostoma elucanum. It has a body and a simple tail. The anterior end will resemble an adult worm with the same number of circummoral spines, and the reproductive organs are still immature. There are many species of heterophids that live in the intestines of fish-eating hosts. The major species are Heterophyes, Metagonimus yucogawai, Haplorchis taichui, and Haplorchis yucogawai. Among those mentioned, let us focus more on the Heterophyes. So the adult worm is elongated, pyriform, and grayish in color. It is small in size and it measures 1 to 1.7 centimeters by 0.3 to 0.4 centimeters. The cuticle is covered with fine scale-like spines and the oral sucker is minute. The ventral sucker is thick-walled, muscular, and lies in the anterior part of the middle third body. The genital sucker of our Heterophyes is situated on the left posterior border of the ventral sucker, and there are two ovoid testes side by side in the posterior fifth of the body. And it also has a subglobose ovary which is anterior to the testes, and a vitellaria with large polygonal follicles in the lateral posterior third of the body. Heterophyes is also known as our von Siebold's fluke. Now let us proceed to the ovum or the egg of our Heterophyes. The egg is light brown in color, it is ovoid in shape, operculated, and it measures 20 to 30 micrometers by 15 to 17 micrometers. A fully developed symmetrical myrocidium is already present within the egg when it is deposited by the adult worm. The operculum fits into the eggshell smoothly, and it does not have an abopercular protuberance, in contrast to Clonorchis and Opistorchis. The treatment for our Heterophyes is also praziquantel. Now let us proceed to Metagonimus yucogawai, and it is the smallest fluke to infect humans. So let us proceed to describing the adult worm of our Metagonimus yucogawai. It is small, pyriform in shape with rounded posterior and tapering anterior end. It measures 1 to 2.5 by 0.4 to 0.75 micra. The ventral sucker is deflected to the right of the midline. The common name of our Metagonimus yucogawai is a heterophid fluke. The two oval testes of our Metagonimus yucogawai are located at the posterior third of the body. The globose ovary is found at the junction of the middle and lower third of the body. And it has also a coarse vitellaria in a fan-shaped distribution in the posterior lateral fields. The next one is the egg of our Metagonimus yucogawai. So the egg is described as minute, ovoidal in shape, and it is operculated with a slight shoulder, and it measures 26 to 28 by 15 to 17 micra, and it is fully embryonated when laid by our parasite. This table shows a summary of our intestinal flukes, and please familiarize the various infos presented in it. And we are done with our intestinal flukes. Now let us proceed to our lung fluke. Paragoniasis is an infection of humans and other mammals by trematodes of the genus Paragonimus. There are 40 known species of Paragonimus, and 6 are reported to cause infections in humans. Paragonimus westermani, or the oriental lung fluke, causes 90% of paragoniasis in Asia. In the Philippines, Paragonimus westermani is also the major species that causes paragoniasis in humans. The only other species in the Philippines is Paragonimus siamensis, which has only been identified in cats. Now let us proceed to the adult worm of our Paragonimus westermani. So the adult lung fluke is reddish brown in color, and it measures 7 to 12 millimeters in length 4 to 6 millimeters in width, and 3.5 to 5 millimeters in thickness. And it resembles somehow a coffee bean. It is rounded anteriorly and slightly tapered posteriorly. The tegument is covered with single-spaced spines. The two testes are deeply lobed and are situated opposite with each other, almost midway between the ventricular and the posterior border of the body. And let us proceed to the ovum or the egg of our Paragonimus westermani. So, the egg is oval yellowish brown and has a thick shell which measures 80 to 118 micrometers by 48 to 60 micrometers and has a flattened but prominent operculum. Opposite the operculum is a thickened abopercular portion. And guys, please don't forget the first intermediate host of our Paragonimus westermani is our Antemelania aspirata, and then the second intermediate host is the Sandathelphosa philippina. And the treatment or the drug of choice for our Paragonimus westermani is our praziquantel. This is an actual image of a prepared slide of our Paragonimus westermani under the scanner objective. And this is how it looks like under the low power objective. This is how it looks under the high power objective. This picture shows the egg of our Paragonimus westermani with some of its labeled parts. This picture shows the different trematode eggs found in zoo specimens of humans. These are the various references used for our lesson. And that is the end of our lesson for trematodes. I hope that you have learned something from this video. Thank you so much and have a good day.